Hello everyone, my name is Eduardo Castro and I'm going to give this presentation which basically explores the analysis of predictive models on brain imaging data, specifically in the context of Huntington disease. This is going to be the outline of my presentation. First, I'm going to give a quick overview of what Huntington disease is. Then I'll explain approaches that we explored for population stratification, both in terms of motor impairment and the rate of cognitive change of individuals with HD. Next, I'll discuss a holistic analysis of brain trajectories by incorporating multiple brain structures as opposed to a single one. Then I'll talk about an approach to integrate multiple diffusion microstructure measures in order to detect patterns of white matter abnormality in HD individuals. And finally, I'm going to wrap this up by providing a summary of our results. But first of all, who are we? I am a member of the Computational Psychiatry and Neuroimaging group of the Healthcare and Life Sciences Department of IBM Research. And the goal of our work is to perform computational analysis of data from a variety of data sources, ranging from brain imaging scans to data from wearable devices and the analysis of speech, both in terms of the acoustic information and the content information of speech, in order to address clinically relevant tasks. We analyze a variety of neurodegenerative and psychiatric conditions, and our work is done mostly as a collaborative effort jointly with clients and collaborators from academia. Now, what specifically is clinically relevant? I'm going to give you some examples. The detection of individuals at risk of developing a disease is one such instance. Another thing that is of interest to us is detect the disease rate of progression of an individual. Understanding what are the biological mechanisms of a disease that are not known at all through the analysis of brain imaging. And finally, the prediction of prospective outcome of an individual. All of these tasks are done through the use of predictive models. So I'm going to discuss a bit what we mean by predictive modeling. As you may know, all models can fit data. Even the extremely complex models can actually fit data. The question is, how well can they generalize? The goal in our case is to test the model's reliability on unseen data, which is basically what is done in predictive models, but that's the goal of our research, which is mainly move from just doing within sample statistics to finding predictive features that are going to perform well on unseen data or unseen individuals. Next, I'm going to explain the pillars of our take on predictive modeling. First, obviously, as I said before, we focus our work on individualized predictions on individuals that have not been seen, rather than estimating within sample statistics. The other important point is that these models need to be robust. And in order to test this robustness, we usually try to validate these predictions across imaging sites and, where possible, across full subject cohorts. And another point is we try to use models that are very low complexity in order to compensate for the high dimensional data at our disposal with a limited number of subjects. And maybe even more important than all of those, if we are able to have a robust model that doesn't give us any information whatsoever of the disease, this really doesn't comply with our expectations. We, in addition to having good performance, we need to get some understanding of what's going on with the model. In other words, we're not looking for black box solution. All of this being said, what is the value of brain imaging? We think of two main takeaways of the value of brain imaging. First of all, it enables the understanding of the neurobiology of the disease, which could be the detection of the location and dynamics of brain atrophy, both in terms of neuronal loss and axonal damage, and also the location and dynamics of neuronal dysfunction. In addition to that, it can allow the early detection of the disease prior to the damage being so profound that nothing can be done about it. Specifically in the case of neurodegenerative disorders, brain pathology usually precedes symptoms. So in early stages, brain atrophy is usually already present and has better signal to noise ratio than clinical markers, which are not as reliable as potential brain markers of disease. Now that you have some understanding of our approach to the exploration and analysis of brain disorders, I'm going to provide some basic information of what Huntington disease is. It is a neurodegenerative disorder such as Parkinson's or Alzheimer's. It is hereditary. The symptoms show up at adulthood, and it affects cognition, movement, and behavior, and it is unfortunately fatal. Also, it is caused by a single gene with an excess of CAG repeats. It can be detected from blood prior to symptom manifestation, and this excess of CAG repeats defines to a great part the rate of progression of the disease. So what is the role of brain imaging in HD? Well, it happens that disease progression is partly explained by the excess of CAG repeats, so-called genetic load, but not completely defined by it. Also, as I told you earlier in this presentation, clinical impairment before diagnosis, while it is present, is really mild and has a limited retest reliability. And this poses the following two questions that I'm going to tell you right now. Can brain imaging be used to characterize HD's pathologic processes? And also, can we infer, based on brain changes, what is going to be the clinical progression of the disease? And these are the two main questions that we've explored in our work with HD, some of which are addressed in this presentation. So these are the problems of interest that I'm going to discuss with you throughout this presentation. First, we wanted to see if we could achieve prospective prediction of clinical impairment with baseline information. Then we evaluated whether we could identify predictive and multivariate patterns of volume change in HD. And finally, we wanted to detect patterns of white matter degeneration across various diffusion microstructure metrics.